Hello, and welcome to another episode of Anything Science. I'm glad you've decided to join me once again for another episode. And for those of you that had liked, commented, and shared in the previous podcast and video, I'm very grateful. The last time we had come around, we spoke about Frederick Mischer and his remarkable journey on discovering the nucleus. This time around, we're going to go a little further and deeper into the nucleus. We're going to discuss the remarkable journey of Walther Flemming and how he managed to unravel the secrets of chromatin and mitosis. So before we get into that, I wanted to lay down a little bit of information that I missed out over the last episode. Something that one of my listeners had pointed out was during the early 19th century, late 18th century, in order to keep the pus cells and the samples that Frederick Mischer had acquired in a condition that it could still be used, considering they were living in the Swiss Alps, was that Frederick Mischer and his lab assistant would keep the windows of the chapel open. And during the winters, through all the snow and the cold, Mischer and his lab assistant would stay snug and wrapped in multiple layers of clothing while the samples were all kept exposed to the absolute blithering cold. That is how he managed to work day in and day out until he managed to acquire the data he needed from all the pus cells. So moving on towards Walther Flemming. Walther Flemming was born somewhere in the mid 1840s, and he was born in Germany. So Flemming had a deep appreciation for education and for the sciences, which kind of helped him in understanding a lot about the natural order of things in life. Frederick Mischer's father, Carl Flemming, was a prominent psychiatrist, and mother was actually quite a huge emotional support for Walther Flemming's childhood. To summarize all of Walter's childhood, I would say it was entirely filled with nature walks and books and a lot of questions about how the world worked. It was a rather curious. He was a rather curious and observant child, and always eager to learn and explore. So this is probably what led him to pursue a career in medicine and further on into research. And I'm sure that's where he believed that he could cover the secrets of life itself. So in 1862, Walther enrolls himself at the University of Rostock to study medicine. And his academic journey was marked by excellence and a keen interest in microscopic anatomy. Obviously, physiology was a crucial element of his coursework at the time. And during this period, Walter's fascination with cells and their inner workings began to take a larger form, a bigger shape in his life. So under the guidance of his professors, he develops a deeper appreciation for the complexities of cellular structures and the processes. But after graduating in 1868, Walther embarks on a journey to further his education and gain practical experience. He works at the University of Prague and the University of Vienna, where he hones his skills in microscopy and histology. These experiences were invaluable to what was going to become a crucial element of the genetics and the research and his research future. So in 1873, Walther returns to Germany and accepts a position as a professor at the University of Kiel. And it's here in the serene surroundings of northern Germany where he would make the groundbreaking discoveries, first through the chromatin and then further. So Walter's work was driven by a simple yet profound question about how do cells divide and pass on the genetic materials to their respective daughter cells. Obviously, at this point, there was no concept of a sister or a daughter cell. It was just how is genetic material passed down. So to answer this question, Walther focuses his research entirely on cell division in animal cells. He meticulously prepares slides of various tissues, staining them with different dyes to highlight the cellular structure. It was quite a painstaking process, which requires immense patience and precision, but Walther was undeterred, and he knew that the answers that he looked for were somewhere within the intricate dance of the cells within each of their inner components. For those of you that are pursuing molecular biology or are pursuing molecular sciences, uh, this is going to be an interesting portion for you, because in your first year or your second year, you will learn karyotyping and chromatin staining. And this is the origin of that very story. So around 1879 comes Walther's first great discovery. And it was this thread-like structure that he observes within the nucleus of the cell. And it is something that kept changing its appearance during cell division. And he names this material chromatin, derived from the Greek word for color due to its ability to absorb specific dyes. Chromatin appears as a dense network of fibers within the nucleus, and Walther was fascinated by its dynamic behavior during cell division. So, through meticulous observations, Walther notes that chromatin played a crucial role uh, in the duplication and replication steps. It was highly organized, with each set um, finding its own way of unweaving itself, unraveling itself in a very organized manner. So the Greek word that he uses to label the replication and the various stages of cell division is mitosis, derived from the Greek word of mitos, which means thread-like. A lot of the details of his work are in the book Cellzubands, Kern und Zeichnung, which translates to Cell Substance, Nucleus, and Cell Division, which was published in 1882. Now, mitosis is often described in the textbooks as the process by which a single cell divides to produce two identical daughter cells. While this definition is accurate, it hardly actually captures the accurate nature of the cell division itself. So every cell in your body contains a complete set of instructions. Your DNA carefully organizes itself into chromosomes. And these chromosomes are like these precious books that hold the code to determining everything about you, from the color of your eyes to the nature of your hair, whether it's curly or straight, to whether you will inherit that single mole on a portion of your face or your arms. Everything is written in that genetic code. And imagine now needing to make an exact copy of the book, ensuring that every word that is written there is passed on. Every word gets passed on. So every word, every sentence is perfectly duplicated during the phases of mitosis, which can be quite challenging, because it can be relayed as a very energy intensive process, and that with today's science and medicine, there is a risk of loss of data. So mitosis in the humans, in the animal cells, or in any living structure, begins very quietly with the soft notes. And I wanted to use this. I wanted to create an analogy that can best describe it. Imagine it as an orchestra, which is tuning itself before a performance. The cell's DNA, which normally exists as a loose, tangled thread, starts to condense, forming visible structures that we call chromosomes. Now, this stage is the prophase. It's almost like the curtain rising, revealing all the main characters of the story. As the cell prepares for division, another extraordinary thing happens. The nuclear envelope, which normally protects the DNA, dissolves, allowing the chromosomes to move freely. It's as if the characters have just stepped onto the stage, ready to take their place for the performance. Next comes the metaphase, where the chromosomes line up neatly along the center of the stage in absolute perfect formation, and they're all poised and ready, each one connected to a set of microtubules that will guide them on to their final destination. Then, with the precision of a well choreographed routine, the real magic happens. The microtubules pull the sister chromatids, the identical halves of each chromosomes, apart, dragging them to the opposite ends of the cell. And at this stage, anaphase, is where the audience might hold on to its breath, watching as the cell ensures that each new daughter cell will receive an exact copy of the DNA. Finally, in telophase, the curtains begin to fall. The chromosomes start to decondense. The nuclear envelope reforms around each set of the genetic material, and the cell itself begins to pinch in the middle, presenting to split by the end of this process. And what was once one cell has suddenly become two, and each with its own complete set of instructions ready to take on its role in the organism's life. Fleming's work, which provided the first accurate description of the phases of mitosis, now known as prophase, metaphase, anaphase, and telophase, which are integral to how the cells reproduce and ensure the continuity of life. Through his observations, Walther identified the mitotic spindle, a structure composed of microtubules that played a crucial role in the movement and segregation of the chromosomes. And his work provided a comprehensive understanding of the mechanisms of life mechanisms of the cell division that ensured the accurate distribution of genetic material during cell division. But obviously, as any other scientist at the time, Walther Flemming's discoveries were as groundbreaking as they were not without challenges. The scientific community was very skeptic of his findings, considering they were still under the clay and sculptor model of genetic inheritance. And they continuously challenged his theories as he continued to challenge the existing ones about cell division and heredity. So, for Walther's meticulous documentation and his compelling evidence, it is that which eventually won over the scientific community, but much after he had expired. Now, Fleming's path to this discovery was not easy, and the 19th century was a time of limited technology in biology. Microscopes, though improvised and quite improved, was still relatively primitive compared to modern standards. And Fleming spent countless hours peering through these microscopes, sometimes under poor lighting conditions. And there is a document by one of his peers who had mentioned, if you came across Walther Flemming in the lab close to his eureka moment, you would notice Walther having stained multiple slides, peering through his microscopes, hoping to catch a glimpse of another part of the cell division, because these were all very slow moving, slow transitioning phases. He would stay glued to his microscope, just hoping to catch another glimpse and another and another. A beautiful symphony for him. For Fleming, he had spent countless hours, and it wouldn't have been easy under these lighting conditions, and straining his eyes to see the tiniest of the details inside the cell. And additionally, staining techniques were still in their infancy. Aniline dyes, which were the ones that Fleming used, were not accurate, were not even perfect, and often required careful experimentation to find the right concentration or the duration of staining to reveal the structures of interest. So Fleming had to carefully balance trial and error in his methods, and this slow, tedious process could lead to the experiment lasting anywhere between days to weeks and in some cases months, offering very little progress for him to show. And another challenge for Fleming was the lack of immediate recognition in his work. The scientific community at the time had a limited understanding of the hierarchy of cells, their heredity, and how genetic material was passed. So although Fleming had meticulously documented all this data, the stages of mitosis and the connection between the chromosomes and inheritance, it wasn't going to be established until the early 20th century. And this meant that while Fleming's work was scientifically accurate, its full significance will not be recognized for another 100 years. So Fleming had faced a lot of intellectual challenges of working in an era where many of the concepts that he was exploring were entirely new. The understanding of heredity and the role of DNA hadn't quite matured. Fleming's discoveries were more about the mechanics of cell division rather than the implications for genetics itself. But it still opened up the avenue for genetics hand in hand with other techniques in molecular biology. And despite the initial skepticism, Walther's work did gain recognition and became a prominent figure in the field of cytology. His contribution laid the foundation of our understanding for genetics and heredity, paving the way for future discoveries in molecular biology. Walther stood dedicated to his research, even in the face of personal challenges, wherein he suffered from chronic health issues, including severe asthma, which often made his life and work quite difficult. But his passion for science and discovery kept him going. and he continued to make significant contributions in the field until his death in 1905. To address Walther Flemming's legacy, I would say Fleming's discoveries of chromatin and mitosis and the entire processes of cell division are absolutely profound, and it cannot be stated less the importance of understanding this. Um, it left a very lasting impact, um, and the levels of data and the detailed descriptions of each of his experiments are phenomenal. And understanding it is key in that era to understand the future of genetics itself, including the identification of the DNA as the genetic material and the elucidation of molecular mechanisms of gene expression. Today, Walther Flemming is celebrated as a pioneer of cytology and a key figure in the history of biology. His legacy lives on in the countless scientists who continued to build on his discoveries and his legacy, exploring the mysteries of life at not just a cellular but at a molecular level. And his dedication to science and his unwavering curiosity serves as an inspiration to researchers around the world. To reflect, Walter's life was a testament to the power of curiosity and to the pursuit of knowledge. The discoveries of chromatin and mitosis revolutionized our understanding of cell division and heredity, laying the foundations for modern genetics. And despite facing numerous challenges, both personal and professional, Walther remained dedicated to his research, driven by a passion for uncovering the secrets of life, which very few were able to succeed at the time. And Flemming remained relatively private about his personal life, so there is not much information that I have to share with you on this podcast, none that I could find that would be worthy of exploring further. But regardless of which, the story of Walther Flemming is just one chapter in the ongoing journey of scientific discovery. And his work on chromatin and mitosis laid the groundwork for a deeper understanding of the molecular mechanisms that govern life. Today, researchers continue to build on his discoveries, exploring the complexities of the genome, the regulation of gene expression, and the intricate dance of chromosomes during cell division. Mitosis, in essence, is not just a biological process. It's a story of continuity and renewal. It's the reason a cut heals, why children grow, and how life persists through generations. Without mitosis, life as we know it would not exist. It's a marvel of nature, a reminder of the complexities and just the elegance of life in its simplest of forms. So as we look into the future, we are reminded that the pursuit of knowledge is never ending, and one requires curiosity, perseverance, and a willingness to explore the unknown. Walther Flemming's legacy is a testament to that very power of discovery and the enduring impact of scientific research. And just to put the world in perspective, around the same time that Walther Flemming has made his discovery of the chromatin, the primary mode of transportation is still horses, carts, and railways running on coal are slowly expanding. Nightfall continued to mean oil lamps, while Thomas Edison had just displayed the first publicly run power station in New York. Louis Pasteur was busy perfecting the vaccine to rabies, which will only come out by the year 1885 for human patients. And the British Empire is exerting greater control over the Suez during its expansion into Egypt through the Anglo-Egyptian War. Messages are relayed through runners, and select few centers of civilization had telegrams available. Claude Monet and Mark Twain are the artists on the rise through their notable works, The Cliff Walks at Pourville by Claude Monet and The Prince and the Pauper by Mark Twain. Thank you so much for joining me on this podcast. I know that it's a bit of a stretch waiting for something like this. It's a bit too niche, but I'm really grateful to all of you that stick on and watch my episodes. Next up, we'll be covering the rediscovery of Mendel's work by Hugo de Vries, Karl Correns, and Erich von Schmack, who independently and not entirely aware of each other rediscover Mendel's law of inheritance. So thank you so much for joining me, and hopefully I will see you all again for the next episode with probably a very different surrounding. And don't forget to like, follow, subscribe, comment, and share. Thank you, and have a great week ahead.